But you know, any medical education, you're going to deal with genes and DNA and that kind of stuff. And how I got myself talked into this was April, and I got to talking about science education for adults. And I volunteered to do something to do with biology. And that's how I got the genetics talk. I figured it would be easy because, you know, we could have our own little DNA experiment. We could get some mixed breed animals, and we could test them, and we could have you all guess and see how good you are. And I've got some data, by the way, as to how well people guess, and we can see if we can beat them. So I figured I could do that, and I could talk about all of the new things that are being done in veterinary medicine concerning DNA and genetics and whatnot. And surely I can fill up an hour. So as I'm going through this, it dawned on me that, well, you know, I don't really do any genetics testing, and I'm a vet. Maybe I've been in Eureka Springs too long, and I'm just practicing in the Stonehenge. I better get on. There's a thing called Veterinary Information Network. All the veterinarians in the United States, I would guess, belong to it. I got on there to look at the chatter to see how much vets were using DNA testing and whatnot, and hardly anybody. So I start calling the specialists at my referral lab, and believe me, my referral lab can do just about any kind of test you can imagine. They don't even offer any genetics testing. So I start calling the specialists that I refer to, and they're not doing any genetics testing. And I thought, oh my God, this is going to be hard. So maybe I better back up, and I better just talk about DNA, how DNA goes from being DNA to making a protein. Yes?
Dr. Pike, why would there be really a need to test for DNA?
Well, I was going to get into that later. That's the problem. You see, by the time I see them, they've got the disease.
Oh.
And that's what my colleagues were saying. Well, yeah, this is cool stuff, and they are doing a lot of things. And there will be practical applications down the road. And there are some practical applications now. But keep in mind, if let's say your dog has got a bleeding disorder, it turns out to be von Willebrand's disease. Well, I'm going to diagnose that, you know, long before I do a genetics test to see if your dog has it. If it's got that disease, it's got that problem genetically. So it's a little bit, when a veterinarian tends to see the animal, kind of plays into what applications that we would use in private practice. Obviously, you know, this would have great implications for breeders, you know, more than for what I do or my colleagues do, except maybe if you're a veterinarian that works with breeders. I don't have very many breeders in my practice. And I'm sure that as time goes on, and there, like I said, there already are some applications for it. We'll see more things coming down the road. Okay, so as I'm thinking about this and what I'm going to talk about, I can't help but remember before I went to vet school and I was just learning about science and genetics. In fact, in the genetics course, I was forced to read that book. Everybody that, I was at Trinity University before vet school, and everybody that went through the biology department had to read that book. And I'm telling you what, that book was extremely significant in my life. And I recommend that everybody read that book. As I've gone through a career in a science-based career, over the years, I've taken on a spiritual bent. And because I came from sort of a non-spiritual background, I've made my way to that through science. And it's my belief that science can be a real spiritual journey. And what I've found in science is that when you get to the edge of science, when you deal with the extremely large, like the astrophysics, or the extremely small, like quantum mechanics, you end up in the same place with a paradox. You all have heard of the situation with light being a particle or a wave. Have you heard of that? The slit experiment? You know, particles and waves are two different things, and they're mutually exclusive ideas, but they're both true. And you find that all over the place in science. So while you look at what nature has been able to designs all this wonderful stuff. And it seems like it has to be directed. You know, that's the other side of the paradox. This guy is the side that talks about evolution being pushed. If you can think of evolution being pulled or pushed, okay, right? Pulled would be like if it's being directed, like by a great architect. Okay, pushed would be like it happens automatically, just because of the laws of chemistry or physics or whatever. This is the guy that gives the best example of how you would explain it when it's pushed. And I don't think that you can find the miracle there until you see both extremes of that argument. So I very much recommend that you read this book. And I'm going to read you a little piece from it at the end of the second chapter called The Replicators. We'll get to that in a minute. So go back to the first slide with the different front lens. One of the things that, and this all relates to genetics, because the things that are changing are the genes, okay? And I just want to point out, one of the things I've noticed as a vet, you know, I'll sometimes have people come into the office and I'll say their cat has got diabetes or their cat's got von Willebrand's disease, and they'll be like, I didn't know cats got that. Almost like if you, or dogs, almost like if you get a blood sample, you're going to get green blood or something. Okay? We're we're related. All of us are related. And I like this. You know, I don't want to get in the way of anybody, but this is a human arm. It's humerus, radius and ulna, carpus, you know. There's a cat's, and they're showing you the same parts. But here's interesting. There's a whale. Okay, and inside its fin, humerus, radius and ulna, carpus, there's a bat. Its wing is its fingers. Some interesting things. Amy and I were frantically looking up. There's a way to quantify how related you are to these animals. You know how that is. You just look at how much DNA you have in common with them. Okay? So, anybody want to hazard a guess as to how much DNA you have in common with a cat? 75?
90%?
90? 90 wins. What about a whale?
Not so much.
97? 85? 80%. I couldn't find a bat, sorry. I looked. I couldn't find it. What about a primate? What about a chimpanzee? 98%. A 2% change in the genetic material, and that's what you get. And you know, you consider a 10% change and you go from a cat to a human. So when we're talking about mutations and evolution that make changes, we're talking about tweaking it, and you get a huge change. Mice, 92%. A fruit fly, okay, that gets pretty far away from human, right? 60%. But here's something interesting, 75% of the genes that cause disease in humans, let me say this again, 75% of the genes which cause disease in humans are found in the fruit fly. So when they start studying genetic diseases in people, fruit flies get them a long way down the road. Isn't that interesting? Okay, go. My favorite. You are related to a banana. Right? You are.
Yeah.
You are.
Yeah.
Okay, not as related as you are to a cat, but you are. Okay, what do you think the genetic similarities between you and a banana is?
2%.
2%? What else? Do you know? 60%. We talked about this last night, and I had no idea. I mean, I didn't know this stuff. I just knew that primates were close, so I started looking into it. And actually, I was doing it with Amy, and we were laughing about it. And then Amy said, "Oh my God, a banana is 60%." I was like, kidding me. some more, this, I kind of forgot what I was going to tell you. I was going to tell you this. This talk is going to be, the first part of it's called Thoughts and Doodling. That's this part. Okay, then the next part's the structure of DNA, how you get from DNA to proteins, importance of proteins, why that's so important. About testing in animals, we'll finally get there, about our tests, and then our drawing. Okay, so go to the ones with the chicken. Okay, this is the chicken, right? Okay, apparently this is not like an embryo chicken, okay? And this is normal, okay? And this is a chicken, that's a tooth. Okay, that's a tooth. The way that it works oftentimes as things evolve is you don't lose the gene. There's a mutation, so the gene is forever turned off. And birds, the original birds had teeth. And the chicken still carries the genes for dentin and enamel in teeth. Still carries some tooth genes, but there was a mutation which occurred 100 million years ago. Two mutations, one, a mutation and a change. One was the loss of the ability to form teeth, and the other was the beginning to build the beak structure, and they happened almost simultaneously. They happened 15 million years apart. So when we're talking about evolution, we're talking, the Earth is about 4.5 billion years old. And it took 1 billion years to get to DNA. Because the first thing that had to happen is we had to get molecules that could replicate. And that took 1 billion years. In fact, if you look at the Earth as being, how old it is as being like 24 hours, life as we know it on Earth has been just a few minutes. So these processes take a long, long time. Okay, so just a few other things in the doodling section. Go to the... People often say, why don't you see any half steps? There's a half step. These are Claridae family catfish that have rudimentary lungs. And they can survive. You can't even really call them lungs. They're structures that they can use as lungs that they have in addition to their gills, and they can survive out of water for a matter of days if they're in a moist environment. What else have we got in this section?
I saw them all naked and afraid.
Yeah. Go back again, is that?
That's the beginning.
Okay. All right, so go to the slide that's got the apparatus in it. That. Okay. All right, so in the book, The Selfish Gene, it was probably, we can look it up, maybe Autumn knows. It was written, I think, in the 70s. I read it in about 1980. And one of the biggest, once you get a self-replicating molecule, things get real easy to explain. But man, getting from simple molecules to the building blocks of life to a self-replicating molecule, that's the hard part. Okay, and back when Dawkins wrote, and he wrote his book, when you read that section, because most of the book is not about that, it's about, actually it was an argument that how in the world could you get altruistic behavior in a tooth and claw world of survival of the fittest, and that's what his book was about. That's why it's called The Selfish Gene. But at the beginning of the book, he talks about just the process of molecular evolution. And they knew some things back then, they didn't know a whole lot, and as I got ready to talk to you about this, I started researching this, and man, a lot has changed since I read that book. A lot of interesting stuff.
76.
Huh?
1976.
1976, I figured, because I read it about 80. Okay, so back when Dawkins wrote the book, and this happened in about the 1950s, they started messing around with how could they get from, this is basically the atmosphere of the Earth, way, way back. You've got methane, ammonia, water, you didn't have much oxygen. You had less than 1% oxygen. We have 21% now. All the oxygen came from photosynthesis when plants came on board. This was an unbreathable environment for us. Okay, so what they did was they put water in here, and they had a heat source. You can imagine volcanoes. Okay, they boiled this water, and it comes up here, and it goes around here into our early Earth atmosphere. Now they also had, and apparently it's pretty ubiquitous, whether you got our atmosphere or the atmosphere back then, you had lightning. So another part of this was running this water through this atmosphere and sending electric current in there. And then it would come down here and it would be condensed back into a liquid, come around here and it would go back into there and it would go over and over, and they'd run it for several days. And an interesting thing happened. And they ran this for several days. They started getting a pink goo, and then they got a brown goo, and they analyzed it. Amino acids, nucleic acids, phospholipids. They may not have had phospholipids with that in there, but basically, this is one experiment, and there have been others since, because now they're messing around with these underwater vents. You know, that's a whole nother bunch of chemistry where the lava's deep in the ocean is coming out. But what they found is it's really not hard to get the building blocks to life just by setting up conditions that were there. Now, you're a long way, though. Let's look at some of the building blocks. Go through here. Okay, those are base pairs, and they're part of what makes a DNA molecule. Go on some more. That's a DNA molecule. We'll come back to that. That's another DNA thing. Go on. That's another DNA thing. DNA. That's DNA stuff. DNA stuff. There you go. Those are amino acids right there. Okay. These are complicated molecules, and they're the building blocks of life, and they come about all on their own when you set up the right conditions. Like I said, the problem, though, is now they've got to get, before you can get the whole ball rolling, they have got to get to the point where they're able to replicate. They've got to be able to copy themselves well. That's very important. Fidelity of copying. They've got to be able to, if they're competing with other molecules that replicate, they've got to be able to do it fast, because the slowpokes aren't going to persist, very long. And they've also got to make some mistakes. If they don't make mistakes, nothing can change. So in the beginning, natural selection is kind of a sub-law of just survival of the stable. What persists molecularly in the environment is what's stable. And what makes something more and more stable, you're going to have more of that. And if you can get something to replicate itself, then you're really going to have more of that molecule. And what's come about lately is they've realized that, in the early Earth, there were a lot of minerals, and these crystals and whatnot have got properties, and a lot of these reactions occurred on the surface of these crystals. So you have trillions of little chemistry experiments happening every second for 1 billion years. And then something happens. You got something that, let's go to the DNA. Okay, go back to that one. One more. This is a stylized version of DNA. So this would be a molecule. And in the way that DNA works, and we'll call this whatever. This is one of those base pairs. We'll call it blue for right now. Blue always attaches to green, and orange always attaches to pink. So you can see that when this thing replicates itself, it makes a negative of itself. So when the building blocks come along and they attach to it, then they make the positive to the first one. This could be DNA or RNA. DNA and RNA are not very much different. This part of the molecule right here is a sugar called ribose, and it's deoxyribose in DNA. It's ribose because it has an extra oxygen in DNA. It turns out that scientists believe that RNA was the first thing to come about. And it's actually because it's less stable in this case than DNA. So in Dawkins' book, these crystals, and oh my gosh, this is so complicated, I don't even know, and it's so far out of my field, but these crystals form, they become important. And then the other thing is that we can't see this happening now, because if you make any building blocks to life, microbes eat it immediately. You have to have conditions for this molecular evolution that existed before life existed, because now life are going to use those building blocks right off the bat. So what they're doing now, believe it or not, geologists are getting into this because they're drilling down into the Earth's crust, and they get down about a mile, they're still finding microbes. They get down below that and they're finding very interesting molecules. They're not DNA or RNA, they're like proto-DNA and RNA. And in fact, in Dawkins' book, he doesn't use the term until later in the book, DNA or RNA. He calls it replicators. Okay, because probably the first replicators weren't DNA or RNA. This is the late model. This is the model that took over the world. This is the model that every single living thing has in it now. So the DNA, it won. But there were probably other replicators that came along first, and they're digging down to get conditions that don't have our atmosphere and don't have microbes to find these molecules to piece together that part of the puzzle, okay, to get to the replicator part. So we're just about done with the doodling section. Over all these years, I remember this last part, and it struck me. This is the last part to Dawkins' second chapter, and I just want to read it because it sort of sums it up, his approach, which is very mechanistic, I understand, and there's more to the story, but this is an interesting way to look at things. So he's referring to the replicators, these molecules that can replicate themselves. Glasses on. There was a struggle for existence among replicator varieties. They did not know they were struggling or worry about it. The struggle was conducted without any hard feelings, indeed without feelings of any kind, but they were struggling in the sense that any miscopying that resulted in a new higher level of stability or a new way of reducing the stability of rivals was automatically preserved and multiplied. The process of improvement was cumulative. Ways of increasing stability and of decreasing rival replicators' stability became more elaborate and more efficient. Some of them may even have discovered how to break up molecules of rival varieties chemically and to use the building blocks so released for making their own copies. These proto-carnivores simultaneously obtained food, quote, other molecules, and removed competing rivals. Other replicators perhaps discovered how to protect themselves either chemically or by building a physical wall of protein around themselves. And remember, DNA, that's what it does, it makes protein. This may have been how the first living cells appeared. Replicators began not merely to exist, but to construct for themselves containers, vehicles for their continued existence. The replicators that survived were the ones that built survival machines for themselves to live in. The first survival machines probably consisted of nothing more than a protective coat, but making a living got steadily harder as new rivals arose with better and more effective survival machines. Survival machines got bigger and more elaborate, and the process was cumulative and progressive. Was there to be any end to the gradual improvement in the techniques and artifices which the replicators used to ensure their own continuation in the world? There would be plenty of time for improvement. What weird engines of self-preservation would the millennia bring forth? 4,000 million years on, what was to be the fate of the ancient replicators? They did not die out, for they are masters of survival arts. But do not look for them floating loose in the sea. They gave up that cavalier freedom long ago. Now they swarm in huge colonies, safe inside gigantic lumbering robots, sealed off from the outside world, communicating with it by torturous indirect routes, manipulating it by remote control. They are in you and me. They created us body and mind, and their preservation is the ultimate rationale for their existence. They have come a long way, those replicators. Now they go by the name of genes, and we are their survival machines. And I've remembered that my whole life. Now that's a very mechanistic way of looking at it. But when you think of the creation as being perfect, a perfect thing would not need, it would be its own creation. It would reflect, if you want to get spiritual about it, it would reflect the Creator. It would happen automatically, I think. Anyway, there's some miracle in that for me. Also, I want to add, I saw an interesting experiment. They're now using evolution to create different molecules that they want. They'll take glass beads, they'll coat them with a chemical that has a certain shape to the molecule of the chemical. They'll take RNA or DNA, and they'll pour it into the flask with these beads. And then sometimes some of the molecules will stick a little bit. And they'll let that happen, and they'll pour the remainder out. They'll take the stuff that's stuck okay, they'll run it through another cycle, and then where it, that's a process of natural selection that they're making in the lab, and they'll do that over and over again, and they'll get molecules that will be a perfect fit. So they'll design molecules using principles of natural selection. They're doing that in the lab, which is fantastically interesting, I think. Okay, we got through that. Has anybody got any comments or questions? Thoughts? No? Okay. All right, so.
I'm trying to process the ant thing.
The ant?
Yeah.
Okay, so go back to the structure of DNA. Another way. Okay, there, about that one. Okay, this is DNA. You can tell because the only difference between DNA and RNA is there's an extra oxygen there. One is deoxy, that's missing one, and the other is ribose, nucleic acid. Okay, so this is the building block to DNA. Okay, and it's got three parts. It's got a phosphate group, it's got a sugar, ribose, either deoxyribose, which would be DNA, or ribose, which would be an RNA. And it's got a nitrogenous base, okay? And there are 5 nitrogenous bases, okay? Go back to the nitrogenous bases. Okay, stop, go to that one. Okay, these represent the nitrogenous bases. Okay, so this is a code made up of 5 different things, okay? So go back, okay, right there. Here they are. Okay, so, when they pair, okay, when they pair, you always get a pairing with thymine and cytosine, which are these two, okay, or, is it right? No, I'm sorry, thymine and adenine, that one. Okay, so those two. And then cytosine and guanine make the other pair. Now you've got this extra one out here. That's because uracil replaces thymine in RNA. So if it's going from DNA to RNA, the thymine won't pair with adenine, it'll pair with uracil. But it still passes the codon, right? It doesn't matter if you read the book Moby Dick in book form or on a disc. It's still passing on the story. Okay, so go to the one slide that I call the enchilada slide. Okay. Okay, so here's this basic story. This is a segment of DNA, and it's paired. Okay, and this represents a big long string of DNA. The code for your body, if you stretched it out, is about 7 feet tall. And of course, remember, it's molecular, so it's a heck of a lot of information. And very little of it actually codes for you. Because in the natural selection, survival of the fittest thing, it's the genes that are competing. Okay, so you've all heard of retroviruses, right? Retroviruses cause AIDS. They cause feline leukemia. They cause feline AIDS. They cause equine infectious anemia. There's all kinds of retroviruses. And what the retroviruses do is a sneaky thing. They've got an enzyme that can go from RNA to DNA, and they take their RNA viruses, and they take their RNA, and they put their information into your genome. So now they put their genes into yours. And your cell goes along, and they use your cellular machinery to make more viral particles. Okay, but when that happens over millions of years, sometimes mistakes are made, and those viruses can't get out. There's been a mutation, and the cells can't make more viruses, but their DNA is still in there. So along the way, through evolution, you pick up a lot of DNA that has nothing to do with making your body. It's along for the ride. It's like parasitic DNA, But in any case, this would stretch out about 7 feet worth of DNA. And a whole lot of these base pairs and a whole lot of code, we consider that's molecular, 7 feet. Okay, so what'll happen is this DNA, a section of this DNA, and there's a whole science, because obviously genes are expressed at just the right time. Okay, so these genes live in a chromosome, and the chromosomes have got protein associated with them, so only parts that are opened up so this can happen will the gene be expressed. But this is a section here that's duplicating itself, so it will open up and it will pair with the building blocks of DNA to make a copy of itself. And that happens during like cell division. Okay? Now, in order to control the body through tortuously indirect means, like Dawkins talks about, you're going to have to make a protein. Because proteins not only are the structure of the body, but they also, enzymes are proteins, and enzymes control the rates of reactions, or whether or not reactions in your body will occur at all. Enzymes which are proteins, can be clotting factors that have to do with how your blood clots. I mean, the whole ball of wax is in protein synthesis. That's the structure of your body, and that's what controls everything. So this is in the nucleus of a cell. Okay, and it splits off now, and then RNA pieces come in, and they pair up and pass the information along. So it'll form a string of RNA, okay, and that string of RNA is called messenger RNA, okay, because it's going to take the message out to the cell to make the protein. It's taking the information from the DNA, it's converting it into RNA, and it goes out into the cytoplasm of the cell. That process is called transcription. Okay, it comes out into the cell, and go to the one that shows the ribosome. There you go. Okay, so this is a string of RNA. It's got its code from the DNA in the nucleus of the cell, and it goes out onto there's all these structures out in the cytoplasm of the cell called ribosomes. And actually ribosomes are two proteins that this goes through like it pulls through like a zipper, and it reads it. Okay, and how it reads it is, this is another type of RNA called transfer RNA. And transfer RNA only always has 3 base pairs on it. So as this comes through there, a piece of transfer RNA which matches the RNA here, it's the complement to it, comes in and attaches to it, and every single one of those transfer RNAs has an amino acid. Remember, the amino acids are the building blocks for proteins. Okay, so as this pulls through here, or as this goes through here, and these attach, and these 3-letter sequences are called codons. Okay, so as these attach, they've got their amino acid with them, and you get this string of amino acids coming out. Okay, that's what a protein is. It's a string of amino acids. And these amino acids can be positively charged, the outside of them, they can be negatively charged, they can be neutral. So imagine a string of amino acids. Imagine a string of pearls that have got a bunch of magnets that are magnetized. You take that, you throw it up in the air, and it's going to all come together in a ball. Well, the way that it comes together has to do with the charges on these amino acids. That forms the exact shape of the protein. If that protein is not shaped exactly the way it's supposed to be, it will not do its job. And that's how disease occurs. Okay, because what happens is the DNA makes a mistake. Show the hemoglobin molecule. Okay, this is a protein molecule. This is hemoglobin. And it looks incredibly complex. But it has to be in exactly that shape where it can't do its job. It can't carry oxygen in your blood. Now, there are sections in this molecule, and I probably could have found a better example, but there are sections in this molecule that aren't where it bends or something. And those sections you could have mistakes in, and it wouldn't change the shape of this hemoglobin. So you could get away with that. And mutations happen at just a regular rate. Okay, and if they happen in a place that's not critical, because it's not where a bend is occurring or something like that, then you'll get away with it. And so, but if it happens in a critical place, where it changes the shape of that protein, then you got 2 things that are going to happen. One, and this is the most likely thing by millions of times, it destroys the protein's ability to do its job, and it kills the organism. But once in a while, it does something that makes it better. And that's how natural selection works. You know, but most of the time, when there's a mutation, it's detrimental. But evolution relies on that mistake that somehow made it a little bit better. Okay, so where do we go from here? Okay. We'll go from here to an example of a couple of animal illnesses. All right, these test kits that we've got there's 2 varieties that you can buy. One is like the ones that we've got, and they test for 250 different breeds. But they don't test for any diseases except for 2 diseases. This comes with actually testing your animal for a genetic mutation called MDR1. And it also tests for a disease called exercise-induced collapse in Labrador retrievers. And as it turns out, that doesn't just happen in Labrador retrievers, but apparently that's where they first found it. Okay, in both of these instances, what's happened is that code had a mutation in it, which caused the protein product of that DNA to not be functional. Now you get, with every protein that you make, you get 2 chances, one from your mother and one from your father. Okay, so luckily for us, most mistakes aren't from both the mother and father for that piece of DNA. Okay, so that's what they mean when they say it's recessive in you. That means you're carrying the faulty DNA, but you got another set from your other parents, so you're okay. Okay, so it's only when you get a defective set from both parents that you're in trouble. In the case of this NDR1, and you know, this is something that comes up actually in practice, because what this problem is it interferes with a protein called P-glycoprotein, and this protein is involved in the blood-brain barrier that stops toxins from getting into the brain. And you know, drugs, a lot of the drugs that we use are toxic at certain levels. So these animals are sensitive to a lot of the drugs that we might use. Now, fortunately for us, we know it's herding breeds that have this in their genome mostly. And the main drug that I use would be ivermectin, which is used in some heartworm preventatives. Okay, so if I've got a herding breed dog, I may not know that it's got this problem, but I know that that problem is in that breed. And so I would shy away from certain drugs. Imodium, you know, like Imodium that you take for diarrhea, very sensitive to that. Acepromazine, which is a tranquilizer that we use fairly often. And then some of the products that we use for heartworms. Now, at the doses that we use ivermectin and some of those drugs for heartworm prevention, apparently they don't cause these animals a problem. But at higher doses it would. If you use them at higher doses, for example, to worm them for something else, it could cause a problem. Nevertheless, I try not to use those drugs anyway in herding breed dogs. But before we recognized this genetic defect, for years I didn't know that it even existed and never had a problem. But that's one of the things that this test kit tests for. The other one, this exercise-induced collapse in Labrador retrievers. My goodness, I'd never heard of it. I mean, a lot of these things are rare. These dogs are fine until they exercise, and then they get wobbly, can't use their muscles, and their temperature spikes to about 107 degrees. You know, if you brought that to my office, I probably would assume that it had a heat stroke. So I probably wouldn't know that it had this problem until after it happened the second time, and I wonder whether or not you didn't learn your lesson the first time with it. And maybe I would start looking into it. It's very rare. I've never seen it in 27 years. But apparently it's becoming more common, or they wouldn't have included it in this test kit. And once again, what its problem is there is a gene called the DNM1 gene, okay, and it codes for a protein that's involved in neurotransmission. And in fact, they even know the site on that protein that got messed up. It's, for what it's worth, it's the G767T site, okay. But apparently that was an important part of that protein, and if there's a mutation there, it's ruined, and these dogs, you know, basically can't exercise. I have a list of diseases that you can get through this company, Wisdom, and there are gave me a number. Oh yeah. $172 genetic diseases that they can look for. Now that's pretty much the same for all these companies. As it turns out, there used to be a lot of companies that did this, and they're all being bought by Mars.
So yeah, that's what it looks like.
So you call them up, and Wisdom is a sister company to Royal Canin, like the people that make the diets.
Mm-hmm.
Okay, well, Royal Canin has got DNA testing, but it's blood tests. Okay, and Wisdom also produces a blood test. I checked on that. There's about a 2% difference in accuracy between these swab tests and the blood tests. And I think the biggest problem with the swab tests, and when we open these up, I didn't want to open these kits up, so we're going to open them up and read the directions. I've never used this swab test to do any genetic testing, so this is going to be a new thing for me too. But apparently the problem is that with the blood test, you're just getting the dog's DNA from the blood. With this, you're getting the DNA from the dog and anything that he ate or got into his mouth. So if you get any banana in his mouth, he's gonna come back retriever and part banana. So apparently when we do this, we can't contaminate the sample. And of course, you could realize that if I took a blood sample, it's not going to be contaminated with anything else. So I think that's where the 2% goes in. You know, and obviously the way that they process the test is going to be different. But one of the things that, you know, I wanted to do, which I found out you can't do, is I wanted to take a sample from, because one of the questions when I was looking up, you know, how this stuff is being used, I was thinking, well, how do you know how accurate this is? I mean, what do we measure this against? So right away I'm thinking, okay, what I'm going to do is I'm going to send one sample to one lab and another sample to another lab. Right. And okay, so what do you think you're going to send? And I had a client do this, and I didn't know this at the time. They sent the same sample and I think a different name. Is that what they did?
They changed the dog's name but sent the same sample to the same company to see if they could tell if it was a different dog.
Okay, well, here's the thing on that. Okay, they may tell you something that in reality isn't true. I don't think they will. I think these are actually pretty accurate. But let's say that they tell you something that's wrong. One thing that's not going to be wrong at all is DNA is a fingerprint. And this company will know if you did that. I mean, they'll catch you right away because they know it's exactly the same DNA. Okay? So, on accuracy of these tests, obviously when we talked about how closely we're related to a banana and certainly other mammals, now we're looking at differences between breeds within a species. So you can imagine that difference is really small. Okay? And then if you look at, let's say, different breeds that are sort of the same variety, like herding breeds. And there's several different herding breeds, right? Well, that difference is going to be even smaller. Okay, so doing a whole bunch of reading and getting a whole bunch of opinions and talking to a lot of people, the consensus that I get is if there's a pure breed in your pet's ancestry to the grandparent level, 25%, you can count that's accurate. So if it says you've got 25% or higher golden retriever in there, you do. When you get below 25%, like if all grandparents were extreme mutts, the accuracy starts going down. And when you get to about the 12% level, you're good. Back when they had more companies doing this, and people were sending samples to different companies, what you'd start to see at the 12% and down level is the companies would get it right and say it's a herding breed, but they might disagree on the exact breed. But once you get above 25%, apparently it's pretty darn accurate.
How does that compare when they're analyzing herding breed, you know, from hunting breeds or whatever? How would that compare in human DNA tests? Like the accuracy for humans, you know, if we're
I think the accuracy for DNA tests is the same for humans and for animals. It's just DNA.
Okay.
That was part of the medical doodling, part of the lecture, that this is all the same stuff and we're all related. I don't know, you know, how this all has to do with how many sites they're looking at along a molecule of DNA. And this company, Wisdom, you know, started out looking at fewer sites and more, and I don't know what they're up to now, but thousands of sites.
I mean, as we travel around the world and we become, you know, some of our kids marry people from Switzerland, and then their kids travel around the world, you know, and marry somebody from England, or, you know, years ago when they came to America, we just kind of stayed in America, you know, kind of like your English bulldogs would stay with English bulldogs, you know what I mean?
Mm-hmm.
And I was wondering, you know, if the more we travel, if we what would you call that, evolve? Would we evolved.
Well, yeah, it is. It's part of that process of evolving. What happens is you get populations that get separated. Okay, and these mutations happen at a very regular rate. I can't tell you what it is off the top of my head, but the people that deal with this know that rate. So example, if they look at this hemoglobin molecule, and you know, remember I said that if it happens at a critical juncture, it'll screw up the hemoglobin and the individual will die. Okay, but when it happens in other places that aren't critical, then it's passed on. So if you separate a population of people, and they're separated long enough, that rate of mutation that goes on all the time starts occurring over a period of time. And the longer they're separated, the more distinctive their DNA will become. And you're right, when you start mixing around all over the place, that becomes blurred out a lot.
So these animals, and dogs in particular, when they have things that are genetic, like we used to have a schnauzer that had pancreatitis, and they told us that the pancreatitis was common in schnauzers.
It is.
You know, they know all that, you know, but.
And you know what? While it is true that pancreatitis is common in schnauzers, and obviously if you're seeing something more in one breed, or more in one race of people or whatever, obviously there's a genetic reason for that. Sometimes we know that there's a genetic predisposition for a problem, but they haven't worked out the genetics part yet. And I honestly don't know about pancreatitis and schnauzers, if anyone's
Is that
done
what
that.
would be, could overbreeding the breed, how does that overbreeding figure in with that, or is that just a term that?
No, it has to do with the problem with recessive genes.
Okay.
Okay, and if you're talking about, and this is where I'm not the geneticist, but if you're talking about the places where this molecule can't change or it won't function, those are called highly conserved areas. That means that they're conserved because if they get changed, you die, generally. I forgot where I was going with that, I'm sorry.
So the more that our dog, had she not been sick,
Okay,
had we
so-
bred her,
Right, so probably, this is where I was going with that. You have a greater probability of a problem if you've got multiple mutations for the DNA that codes for that protein. Okay, and if you start messing around and you have relatives mating with relatives, you're setting yourself up for more probability of that happening.
okay.
Just like if you know, when they talk about classical genetics, where they're not really talking about DNA, but they're just looking at generations and pairings. And you know, in school they always talk about hemophilia.
Yeah.
You remember that in biology? And what do they use? They use the royal family because they were constantly intermarrying. Okay, and then increasing the likelihood that recessive genes would, you know, that you'd get genes from both sides of your family because you're marrying your cousin or whatever.
So you were saying earlier, like if you sent the same dog's sample to two, you know,
Right.
under two names, what if, okay, you know the dog's mother and father for certain,
Mm-hmm.
okay, and there are multiple puppies,
Mm-hmm.
and what if you had different siblings from that litter tested? How close are they gonna be?
Too close to who? The
To
mother and
each
father?
other, to each other. Because you were saying that
They're going to be very
that it's a
close.
fingerprint, so to speak.
Well, not
So
that
it's a
close.
little different.
Not that close.
Right, but, so they're gonna be slightly different.
You're going to be the closest to your sibling. You're
Okay.
more related to your sibling, obviously, because
Right.
you've got your mother and father's genes.
Right, right,
Okay? And then you're second most related to your mother and father.
right.
But the gametes, the sex cells from your
Right.
parents aren't genetically the same,
Okay.
exactly the same.
So,
So you don't get the entire complement from your father.
so in this ancestry.com stuff craze where people are sending in their own,
Mhm.
you know, testing, mine won't necessarily be the same as one of my sisters, but it'll be very close. My sister or my
It'll be the closest to your sister than your mom. Now, and once again, I'm getting out of my field here a little bit,
Sure.
but this stuff, they're figuring out to the point, you know, it doesn't matter whether you send your genetics in, if enough of your relatives did.
That's kind of what I was
And that's the deal. I mean, they're catching people that have committed crimes, not because they ever turned their DNA in, but enough of their relatives did. Their cousin did, you know, this person did, and that person in their family, and they have gone looking for the person.
That's how they found that, they're murderers.
Right.
Oh,
That's how they
so
caught, didn't they catch the BTK killer that way?
Yeah.
He never turned his DNA in. But enough of his family, you know, thought, oh, this is cool.
They got them
Then
cousins.
they got him anyway. Yeah.
Okay.
So, I mean, I can't
Yeah,
give you
that
the,
tells me a lot.
I can't give you the particulars, but I mean, you
That
know,
tells me a lot.
that's how, so if they can do that, don't try to send in the same sample from the same animal, they'll know right away.
Well, I mean, I have a friend who sent off for her,
Hmm?
and
Did you
they-
touch that friend? No.
they sent her back information,
Uh-huh.
and she found siblings she never knew she had.
Oh, that's happening.
But they sent her that information somehow. That's how she discovered them somehow. Or they sent it.
Email to you off from your DNA.
Yeah. And so she has discovered now 3 siblings when she thought she was an only child.
Yeah, and I don't know how that exactly happens, but when I was looking through this stuff to try to figure out what to talk to, I was running into that all over the place, that people are finding out they have siblings they didn't know that they have. Yeah, and somehow or another, the company that's doing that's tipping them off to that.
Yeah, right. Add a little bit to that.
My understanding is that when you send your sample in, if you read the fine print, it says that that information will be public. And they cross-reference with computers that can go through a lot of information very quickly.
Right.
And they send it off to whoever, had originally sent it. And with finding the people who had murdered other people, they do a layout of all the people that show up.
That you could possibly be
Right.
related to.
And
Yeah.
they look to see generally who's still alive, who may have died when, where are they located. And they use public information for that. And they can narrow it down to a very small amount of people, if not just 1 or 2. A detective then starts following that person and waits for that person to throw down a cigarette, throw away the cocaine, and they run it.
This is,
There has not,
go ahead.
there hasn't been a challenge to the Supreme Court about privacy about that yet. Some law enforcement people expect that in the future there may be, but because the baseline test, when you send it in, the fine print says that that information will be publicly available, there has not been a challenge to it yet.
And this is a really interesting thing too, because this is where genetics meets computerization.
Sure.
I mean, you know, they've got this vast amount of data out there that everyone's given them, okay, and now they've got computers that can sift through information. You know, if this were decades ago,
This
they
was
might have,
scientist at a bench, you just couldn't go.
yeah, it would take forever to go through all that data, but they're putting that stuff into databases that are searchable. You know, I mean, I think we all need to think
So
about,
I think that's what happened with my friend, is it was put in a database and another sibling contacted her.
yeah, and they're racing towards getting it all into a database. I mean, the insurance companies would love that.
Oh, sure.
I mean, that's one of the things that we need to think about is, you know what pre-existing condition is.
Yeah.
You're covered for everything except what may ever happen to you. You know? And, I mean, we need to think about that seriously. I mean, it's not that I'm wanting to get away with the murder, but I'd like to be able to have insurance.
Going back to the nuts and bolts level, your hemoglobin.
Mm.
All right, you've got a replication process that gets a glitch in it, and you get a mutated hemoglobin cell that won't accept oxygen.
Mm.
That's one cell. How does that affect?
No, it's every hemoglobin that's made for that individual. Because the mutation occurred at the DNA. So every time that it,
But where did that mutation occur? At one one cell in the individual?
No, it
How
happens
does
at the
get-
level of the gamete when you were,
So then the.
How
you
do you
never
become
did get,
global?
So
if it was a dramatic enough problem, you never survived.
Yeah.
So
Because
perhaps
the
it was a
mutation happened
miscarriage
in
or
the
something.
sperm cell
Yeah,
or the egg cell,
Ah.
and then you never got to be a
Okay.
person.
got
Okay, that's what it happened, because you're getting a part of your father's and mother's genome, and things can happen, and there could be radiation that damages sex cells, different things can happen. So you never got the opportunity to be a person. Though you can also be born with defective, obviously, proteins like these diseases. I mean, when I look through this list of diseases, one thing is, my God, I haven't heard of half of them. And most of, or many of them, you wouldn't breed the animal, and it's not gonna live very long.
I read something very interesting the other day that hadn't occurred to me. The diseases of aging, eyesight going, Alzheimer's, heart disease, are immune to evolution because they don't present until after your reproductive years.
Let me think about that. I'll have to think about that. That sounds right at first. Unless elders give some kind of selective advantage to the younger ones. You know, one of the problems with Dawkins' approach, because you know his big thing, and biologists' big thing right now, is that natural selection occurs at the level of the gene. Okay, well, I think that's extreme, because obviously what's good for the individual is good for the gene, and what's good for the species is good for the individual. So Dawkins has gotten, like, he's fallen in love with how well this works at the genetic level, to the point where you've even got things, like I said, parasitic genes, okay, where the genes are competing. But you know, I think this operates at more than one level. And that gets into kind of the spiritual side of things, but I think, you know, I don't know what selective advantage to the offspring the parents or the grandparents might bring. I don't know. But I think I think that's right. It may be that, obviously, it's not gonna have as big an
It won't
effect
affect your
if you're not
reproductive
reproductive age, right? And also,
Wouldn't you still have the susceptibility, but it's more like environmental factors that would determine whether or not that would come into play?
for
Like if
Alzheimer's,
certain genes would get turned on or off,
say that again.
wouldn't you still have a susceptibility to a disease regardless of when the reproduction would occur? You'd still have a susceptibility, but there would be a difference depending upon exposure to, say, environmental toxins, for example, and that would turn a gene on or off. The nature and nurture.
Oh,
not-
absolutely. I mean, you can't look at disease as just being genetic.
Right.
I mean, a whole lot of it's environmental, sure, diseases are. And sometimes genetic predispositions just make you more likely to be susceptible to an
Right.
environmental effect.
Right.
So in the real world, it's an interplay between genes and the environment.
Yeah.
And the other problem with these kinds of things, you know, how they basically do it is they look at a bunch of individuals, people or animals that don't have the disorder, and a bunch of ones that do, and then they start looking for differences in the genome. And some things protect you. And as I'm sifting through all this stuff, trying to think about what I was going to tell you tonight, I'm watching a bunch of medical people arguing about the usefulness of genetic testing, and there was a physician talking, and he was saying that these companies sometimes they make some pretty severe claims that they found just the right differences that they can say this person or this animal is more susceptible to the disease. But he had a patient where they had sent this off, and you know, now these test kits and things are available to you, not through any kind of a medical situation. And this person had found that they had some gene that made them more likely to get Alzheimer's. And then that upset them. After a while, they went in and they looked into it more extensively with the medical people, and that was true that they had that gene, but they also had a gene that was protective, and they canceled each other out. The only problem was this poor person suffered for a couple of years, worrying that they were going to have Alzheimer's.
That's just having that knowledge.
Yeah, and so this was a physician talking, and his argument was, you know, we've gotta be careful, we're letting this stuff straight out to the public, and the other thing is, especially the stuff that goes to the public, is proprietary. These wisdom people don't want you to know exactly where these markers are, because they're competing with other companies that might put a product out there. This is also happening with products that go to you for your genetics. And we gotta trust them as far as, you know, if they say you've got a susceptibility to some illness, that they've actually proven that, and we can't always look at their work real well because of it being proprietary. And that gets into a whole bunch of legal stuff. You know, which exact tests and labs can be used as legal evidence, because if we don't know the process that you went through to determine that these markers actually do set you up to have a problem, then that gets into a shifty situation with the lawyers. You know, that's way out of my field, but I mean, I saw some interesting stuff. Even with animals, you know, obviously, I happen to think that, for example, pit bulls can be really nice. I've known a lot of pit bulls. I mean, I don't have a problem with pit bulls, but some people do. You know, and if your landlord does, and you know, you send your dogs testing off and it comes back with pit in it, you know, or let's just say that your dog bites someone after you test them and you know it has pit in it, someone might try to make the argument that you knew. You know, it's kind of like if you come into my office and your dog bit someone, I don't want to hear it.
Yeah.
Because if you ever end up in court when it bites them again, you know, I could just see myself, well, you even told your veterinarian this was a vicious dog. You know, so I know.
So Tony, you said that you never had a client ask you to do genetic testing, did you say that?
No, because this has been straight out to the public for, you know, I mean, this has been released to the public. I've had clients do it,
Right.
and I've told them to go get the test kit, they've got cheek swabs and do it, but there's no, unless you want a blood test run,
Right.
there's no reason to have me do it. Now, there is one other slide I wanted to show you, the one with PinGen. Okay. Now this you would need before. Okay, this is the ultimate DNA group, research group at Penn State, right. And this is their website. You can go there and you can hit tests, PinGen tests. Let's say you own a dog of a certain breed. You can actually search it for what they can look at or find based on the test. But more commonly what you would do is you'd go there and you'd hit the animal, the breed you have. Okay, and it will list what they are looking for in that breed in the way of diseases. Okay, so if you had a purebred animal and you were concerned about having some kind of genetics, and I could bleed it for you, and if we found that they were looking for that test, this is where you would go. I went to this website the other day and played around with it, just putting different breeds in. In some breeds I'd find 5 or 6 different things that they were looking for in that breed, and some breeds there'd be none. You know, but if you have an interest in a breed and what genetic tests that they're running for that breed, and I mean, there's, like I said, there's
The breeders, the dog breeders are doing more and more of that.
Yeah.
And because I was raised by a dog breeder, and somebody who showed dogs there, and I watch a lot of it in the corgis, because and they look more and more at that. And someone will look and say, okay, and I get that they're looking at keeping the breeds more pure and looking at not passing on those things that are really bad, you know, but, you know, the corgi breeders are checking eyes and hips and things like that so they don't pass on hip dysplasia, and I get that, you know, and the newest thing with corgis is testing for degenerative myopathy, and it's bad. I get it because I had a dog with it. But the fact is, it doesn't present till after 10.
right.
Awesome.
Well, you can go on
Yeah.
there, for example, to corgi, and I guarantee you, if they're running a test on corgis, it's these people.
It's there, yeah.
It's on this website. That's the one-stop shopping for animals, for genetic stuff. And you know, you talked about keeping a breed pure. The one time I did work with a lot of breeders was a different practice, and it was cat breeders. And I mean to tell you, The problem is that the ones that were winning at shows I knew had heart murmurs and things because I was their vet. And nobody checks them for, or they do now in some breeds, but in some breeds they don't, you know, whether or not they're healthy is secondary to the way that they look or whatever.
And probably the word I should have used was pure, but they're wanting to breed healthier dogs. You know, in getting and trying to not pass on some of those big genetic faults.
And they, you know, nature does a pretty good job of designing when you're doing it because you're purposely breeding. You know, breeders, they call that line breeding.
Right.
Okay, by another name, it's inbreeding. Okay? And no matter what, when they're keeping a breed pure or whatever, they're setting themselves up for more problems, no matter what.
Absolutely.
And so as they go on and a breed's been around longer, you know, as a matter of fact, the more popular a breed gets, if you're a vet and you've been practicing long enough, if that breed becomes popular, it just about gets ruined.
Please don't send me Cocker sticker.
Yeah.
That becomes the most god-awful dog on the planet.
So some of them are arguing about, you know, when they've gone down for a whole bunch of generations, maybe we should throw some other breeds in here.
To sprinkle them up a little
Just,
bit.
yeah, just because we're, you know,
Like more robust.
and any of these weirdo genetic diseases, you know, when you run across, or really any defect that I see that's a congenital defect, it's almost always a purebred. You know?
Absolutely.
I mean, you know, I
Absolutely.
couldn't make a living if I didn't have purebred animals out there to break down. You know?
Absolutely agree.
So with animals, it does the same thing with people. I lived in Wisconsin and I knew a lot of Amish people.
Right?
They were giving birth to kids with cleft palates, and many of those kids had one eye one color and one eye another color. So what they were doing was bringing people from around the country, other Amish people, to change things. So they even knew, and that was 30 years ago.
There is interesting stuff out there of concern when a population of people are not getting enough new genes into that population that causes a problem. And you do see that, especially in some religious communities where they only marry and they're kind of isolated. I mean, they are finding problems that, you know, you just don't see with any frequency in the general population because they are doing what the breeders are doing. You know, not to bash breeders, but I mean, we have to be careful when we're breeding. Nature's pretty ruthless, but when nature does it, it tends to keep things strong. When we do it, we're taking over for nature. We may not be as good at design.
Thank you.
All right.
Is there gonna be a test?
Oh yeah, the test part. We were just kidding about the test.
They're just empty
All right,
boxes,
so-
there's nothing in here.
So, who's gonna drop?
Do you want to draw, April?
I think you should draw because
I
I'm
should draw?
yeah, because I'm I'm holding this.